Pyrimidines, so this is what? So this is specific to DNA, methyl group. So if a radio label, maybe a methyl group or one of the carbons, so that will be highlighted in the DNA or radio labeled in the DNA. So these are pyrimid. Nomenclature of the bases, again, you don't have to memorize. They don't go into chemical formulas anymore. We're all doctors, not going to be becoming PhD students, right? So don't worry. Only few things. The first one is adenine. Adenine is nothing but 6-amino purine. We all know why 6-amino, because amino group is attached at the 6th position, right? Guanine is 2-amino 6-oxypurine. Amino group is attached to the 2nd position. All right, 2-amino 6-oxypurine. For all practical purposes, you need to remember that if you knock off the amino group from adenine, which will give you hypoxanthine. And if you knock off the amino group on guanine, it gives you xanthine. So basically, they are the breakdown products. When you break down adenine nitrogenous base, you get hypoxanthine. And when you break down guanine, you get xanthine. So that is the important. So we have different enzymes which help in breaking down the purines. We'll talk about that in nucleotide metabolism. We are not getting there. So always remember, adenine undergoes deamination to form hypoxanthine. This is important even for molecular biology. Why am I telling you is, we do have bases which undergo spontaneous deamination. Like when exposed to heat or sunlight, there is some amount of deamination which happens in our bases. Adenine, when exposed to